Hi, my name is Melina Morales, and the first question is discussing the polymerase chain reaction. The purpose of PCR is to replicate DNA outside the body or in a test tube. This process allows you to go from one DNA molecule to billions in a few hours. Amplifying DNA through PCR is useful in many situations. It is used a lot in crime scene investigations to have the samples of DNA so you can perform a variety of tests on. PCR was also used in the Ebola outbreaks in 2014. The amplified genetic material was used to determine that different strains had actually caused the outbreaks. This technique is performed with a thermocycler, and the process involves three steps: denaturation, priming, and extension. First, denaturation heats the DNA up to around 94 degrees to break the double-stranded DNA's hydrogen bonds. Once that has occurred, the molecule is primed. DNA primers bind to the genetic sequence. DNA polymerase replicates the daughter strand, and nucleotides continue to build onto it. PCR uses a non-heat sensitive DNA polymerase called Taq polymerase because the temperature reached is really high. The molecule is also cooled to around 65 degrees to reform the double-stranded DNA. The last step is extension. This is where the temperature is raised to 72 degrees. This causes an increase in the rate of replication exponentially. The first cycle produces 2 molecules, the second produces 4, the third produces 8, and so on. After 30 cycles of PCR, more than 1 billion copies will be made. The second question addresses the similarities and differences between viruses, viroids, and prions. First, they are comparable because they are non-living and infectious. Viruses can be either DNA or RNA, but not both, and infect all organisms. Its extracellular state is complete and known as a virion. The nucleic acid has a protein coat called a capsid shell. Some are also enveloped by a phospholipid membrane. Viroids differ in the fact that they are only infectious to plants and lack a capsid shell. They only possess single-stranded RNA as well. Finally, prions don't have nucleic acids, but they do have protein. They are made of cellular PRP protein, which contains alpha helices. When the prion PRP misfolds, it turns to its disease-causing form with beta sheets. They can cause the degeneration of neurons in the brain of mammals. An example would be BSC, aka mad cow disease, or the human form, which is VCJD. Bacteria are complex living microbes that can have both DNA and RNA. That is how they differ from viruses, viroids, and prions. Question 3 addresses the chemical methods of microbial control. The 3 chemical methods that would be most effective against gram-positive bacteria would be surfactants, phenolics, and alcohols. Gram-positive bacteria lack a lipopolysaccharide layer, which makes them susceptible to chemicals. Surfactants are low-level affecting chemicals like soaps and detergents. They have hydrophilic heads and hydrophobic tails, which makes them good for degerminating surfaces, but not for killing the actual bacteria. However, with the use of water, you can wash away the bacteria that was degerminated. Quats or quaternary ammonium compounds are good for disrupting cell membranes. Next are phenolics. These are low to intermediate disinfectants that denature and disrupt cell membranes. They are effective around organic substances like saliva and vomit, which makes them extremely useful in healthcare settings. Lastly, alcohols are also an intermediate disinfectant that denature and disrupt cytoplasmic membranes. They are more effective than soap. However, they remove the good bacteria as well as bad bacteria on skin. These 3 methods would not be useful against bacterial endospores because these chemicals are effective on the cellular membranes of low to intermediate level microbes. Bacterial endospores have multiple thick protein coats around their nucleic acids. You would need a high-level germicide to kill these microbes since they are resistant to many chemicals. Question 4 addresses the drugs that inhibit microbial growth from 4 categories. The first category would be inhibition of cell wall synthesis. This occurs when beta-lactams interfere with the cross-linking of NAM subunits. Vancomycin would be an example of this because the drug works to block the bridges that link NAM subunits in gram-positive bacteria. Inhibiting protein synthesis would be the second category. Drugs that aid this process would target parts of translation. Tetracycline is an example of this. It works to block the A site of the ribosome and blocks the docking of new tRNA/amino acids. The next category would be the disruption of cytoplasmic membranes. Drugs like amphotericin B attach to ergosterol molecules found in fungal membranes, form channels, and damage the structure. Humans are susceptible because cholesterol is similar to ergosterol, but bacteria aren't because they don't have sterols. Inhibition of DNA/RNA synthesis is the last category. Nucleoside analogs are used to block DNA replication or mRNA transcription. We see them mostly used against cancer because of how fast cells divide. Acyclovir is an example of a nucleoside analog that inhibits replication because it so closely resembles guanine in the nucleotide sequence. This drug is used to treat viruses like herpes, cold sores, and chickenpox. Question 5 addresses virulence and virulence factors. Virulence factors are produced by microorganisms and contribute to the severity of the pathogen. The first one is adhesion factors, and it contains 2 parts, specialized structures and attachment proteins. Examples of these would be hooks seen in helminths and ligands on bacteria to bind cell membrane receptors of host cells. These are a necessity to create growth within a host. Without these attachments, the microorganism is classified as avirulent or not infective. Extracellular enzymes are another factor that help pathogens thrive within a host. They work to dissolve structural chemicals in the body. For example, hyaluronidase digests hyaluronic acid, which holds cells together. Collagenase then dissolves collagen to allow entry of the bacteria deeper into the body. Toxins are another factor that harm tissues and trigger an immune response to them. There are 2 types, exotoxins and endotoxins. Exotoxins are secreted by pathogens to destroy host cells and interfere with metabolism. The body produces antitoxins in response to bind and neutralize the toxin. Endotoxins are the waste products of phagocytized gram-negative bacteria cells, known as lipid A. In response, the body triggers fever, inflammation, diarrhea, etc. Lastly, anti-phagocytic factors aim to prevent phagocytosis within cells. Bacterial capsules are made of polysaccharides, which can be slippery and difficult for phagocytes to engulf. Anti-phagocytic chemicals work to prevent the lysing of the phagocytic vesicles as well. On top of this, leukocidins also destroy white blood cells. The normal flora can produce virulence factors when microbial antagonism occurs.